Hi, Mr. Wartsky here again, and this is the second screencast in the energy series. Now, this is the last slide that you saw. We didn't discuss it, so we're going to jump right in and talk about glycolysis as the first step in aerobic respiration. Now, remember, aerobic respiration means that you are producing energy with the use of oxygen, although we will not see oxygen until the last step of the electron transport chain. So glycolysis is the first main set of reactions, and they occur in the cytoplasm. So this is how prokaryotes produce their energy through a process that is very similar to glycolysis. We'll talk about that later. But in cytoplasm, we're going to take glucose, and eventually we're going to change it to pyruvate, 2 ATP molecules, and 2 molecules of NADH. That is glycolysis, the overall. So let's go over the steps. Now, I want you to get the different steps down with the description as well as the enzyme. So pause this as you see or as you think you need to pause it. There's going to be a lot of information here. So the first step is that glucose enters the cell, and it enters the cell via facilitated diffusion. Parts of glucose have a charge. It will not be able to move through the hydrophobic middle layer of the cell membrane, so it needs to have a protein tunnel. That is glucose entering. Now, as glucose enters, it will turn around and head right back outside, except for this second step. So glucose, we're going to have a phosphate from ATP added to glucose, and that does two things. It adds energy to glucose, so it energizes the glucose molecule, and it changes the shape of glucose, and it keeps glucose in the cell. So we have now a molecule called glucose 6-phosphate. Glucose has 6 carbons, and the phosphate is located on the 6th carbon. That's where we get the 6 from. So glucose 6-phosphate, or they can say glucose phosphate, but it's on the 6th carbon of glucose. We also end up with ADP because the phosphate from ATP is added to glucose. Now we know that all enzymes that move phosphates are called kinases. This enzyme is called hexokinase, hexo because of the 6 carbons of glucose. So hexokinase takes a phosphate from ATP and adds it to the 6th carbon of glucose, and we get glucose 6-phosphate and ADP. The third reaction, we take glucose 6-phosphate and we're going to change it into the isomer of glucose, which is fructose 6-phosphate. It's an isomer. Remember, same molecular equation, just a different shape. And the enzyme that does that, it's a great enzyme name because it tells you exactly what it does, is phosphoglucoisomerase. It changes glucose 6-phosphate into an isomer, which is fructose 6-phosphate. Pause it if you need to. Now, this next reaction is an example of reaction coupling. So fructose 6-phosphate is very unstable and wants to revert right back to glucose 6-phosphate. And if that were to happen, then the reaction or glycolysis would grind to a halt. Everything would stop. So right after glucose 6-phosphate is formed, a phosphate from ATP is added to fructose 6-phosphate. We're adding energy to it, and as we add energy to it, we make it a little more stable and keep the reaction moving in one direction. It adds the phosphate to the first carbon of fructose. That's why it's fructose 1,6-diphosphate. And the enzyme, we know that it needs to be a kinase. So it is phosphofructokinase that adds the phosphate from the ATP to fructose 6, the first carbon of fructose 6-phosphate. So it becomes fructose 1,6-diphosphate. Keeps everything moving in one direction. So far, what we've done is we've used 2 ATP molecules. We are now in an energy deficit. Right? We've used it. So in order to produce energy, we have to invest energy into the process. Fructose 1,6-diphosphate is cut into 2 molecules of glyceraldehyde 3-phosphate. So there's another step that I kind of skipped. Don't worry about it. Everything comes out okay. If you want to add more steps, you can look it up. But fructose 1,6-diphosphate is cut in half by an enzyme called aldolase. Aldolase splits it into glyceraldehyde 3-phosphate. We have 2 molecules because each glyceraldehyde 3-phosphate is 3 enzymes or 3 carbons long. So we have two molecules of 3-glyceraldehyde. We don't need the 3-phosphate at the end. And what we're going to do is we're going to add an inorganic phosphate to the glyceraldehyde 3-phosphate. You might be asking yourself, where does the inorganic phosphate come from? It comes from the environment. For us, it comes from our diet. This is one of the reasons why we need phosphate, because we need to add it to glyceraldehyde 3-phosphate. Now, what happens is that 2 electrons from the phosphate are removed and given to NAD, as well as a hydrogen ion, and that forms NADH. When this happens, the inorganic phosphate can bind to glyceraldehyde 3-phosphate on the first carbon. So now we have 2 diphosphoglycerate molecules, 1,3-P, or 1,3-DPG. Same thing. And an NADH. So, it should be 2 NADHs and 2 phosphates. There should be a 2 here, put a 2 here, and a 2 here. We end up with 2 there. That is my fault. The enzyme, it has to be a dehydrogenase because we're moving electrons in the hydrogen ion, and it's triose phosphate dehydrogenase. Beautiful name, isn't it? I know. So again, we're taking 2 electrons in a hydrogen ion from a phosphate, adding it to glyceraldehyde 3-phosphate to form NADH, as well as diphosphoglycerate 1,3-P. Are we done yet? Not quite. Almost so. So we're taking 2 molecules of diphosphoglycerate, and we're going to add ADP. Now, this is a bellwether event right here, because what we're doing is we're going to move a phosphate from the DPG to ADP. So we can get 2 molecules of phosphoglycerate 3P, 3PG, and 2 ATP. Why is that so huge? Because we're actually producing ATP from this step. Great. The problem is we're still at 0. So we've done steps 1 through 7 for absolutely nothing. We know that enzymes that move phosphates are called kinases, and this kinase is called phosphoglycerokinase, and that's what removes the phosphate from the first carbon of diphosphoglycerate and adds it to ADP. In step 8, the only thing that we have left is that last phosphate. And the last phosphate is going to eventually be added to ADP to form ATP. But the problem is there's too much energy holding the phosphate onto this molecule right there. There's nothing that can pull that phosphate off just yet. So what we're going to do, because of the second law of thermodynamics, we're going to decrease the potential energy of that bond by moving the phosphate over 1 carbon. So we move it from the 3rd carbon to the 2nd carbon by an enzyme called phosphoglyceromutase, and that lowers the potential energy of that last bond to the phosphate in the carbon. We have 2 molecules of phosphoglycerate 2P. So on the 2nd carbon, we're going to take away water to reduce the potential energy even more. And the enzyme that removes the water is called enolase, and we form this molecule called phosphoenolpyruvate, which has to be one of my favorite words in biology. It just flows off the tongue, phosphoenolpyruvate. I was going to name my daughter, give her a P at the beginning of her name, because I love phosphoenolpyruvate so much. So we're going to take 2PG, take away water, we're going to form phosphoenolpyruvate, and the enzyme is enolase. Number 10 is this is the thing. This is what we've all been waiting for. We take 2 molecules of PEP, which is phosphoenolpyruvate. We add 2 ADP. Guess what's going to happen? The phosphate is finally going to come off of phosphoenolpyruvate. It's going to be added to ADP to form 2 molecules of pyruvate and 2 ATPs. And you know that if you move phosphate, it has to be a kinase, and the enzyme is pyruvate kinase. So what we have is a net gain of 2 ATPs. We also have 2 NADHs and 2 molecules of pyruvates. Now in prokaryotes, the reaction would stop right here, right? The 2 ATPs is enough for prokaryotes to survive. They're happy as a pig in poo in order to have those 2 ATPs, but we need to recycle the NADs. Because if you run out of NADs, there's no way to receive the electrons in the from phosphates. So we have to recycle them to produce NAD again. We have to change the NADHs to NAD. So how does that happen? Well, that can happen one of two ways in prokaryotes. And that's called fermentation. And we'll get that in a second. This is the overall reaction of glycolysis. Isn't that beautiful? See right here? I skipped this step right here. But see, this molecule is changed into that molecule anyway, so it really doesn't matter. So anaerobic respiration is basically glycolysis. We call it fermentation. But it happens without oxygen, and you produce only 2 ATP molecules. So the pyruvate can go one of two ways, depending on the prokaryote. They can't switch when they want to. We can't switch when we go through anaerobic respiration. We only go one way. You can either go from pyruvate, this is the first one, this is called alcoholic fermentation, in which we take pyruvate, we convert it to acetaldehyde by removing carbon dioxide, take away CO2, gives you the bubbles, right? And the enzyme that does that is pyruvate decarboxylase, removes the carbon. We have 2-acetaldehyde, and this is the step that's important. The 2 electrons and hydrogen ion are removed from here and added to acetaldehyde, and that will form 2 molecules of ethanol and NAD, which goes back to, I think, step number 6 to remove the electrons and hydrogen ion from the phosphate. The enzyme that does this is alcohol dehydrogenase. Yeah. And we have formation of ethanol, which is drinking alcohol. Yay, this is actually alcoholic fermentation. This is what happens when you produce alcohol. Woohoo. And this is it right here. The second form of fermentation is lactic acid fermentation, in which you don't produce ethanol. Instead, 2 pyruvates, you have 2 electrons and a hydrogen ion from NADH is added to the pyruvate to form lactic acid, and the enzyme is pyruvate dehydrogenase. Now, we don't go through alcoholic fermentation when we do anaerobic respiration. We go through lactic acid respiration. That's why our muscles feel the burn, because of the buildup of lactic acid. If we did produce ethanol, then more people would be addicted to exercise. Instead of feeling the burn, they would become intoxicated at the gym. They'd have to have breath analyzers before they left gyms. And here is lactic acid fermentation. So if you're a prokaryote, you'd either do lactic acid fermentation or alcoholic fermentation. If you're going through anaerobic respiration, for us, we would do lactic acid respiration. If you're a lactobacillus acidophilus, you do lactic acid fermentation. And I'm sure we'll talk about that in class. So we're ready for the mitochondrion, and this will be the last thing for today. It has 2 membranes. We're going to draw the picture. It has an outer and inner membrane. The inner membrane, 80% of it is proteins of the electron transport chain. They're found in folds, and those folds are called cristae. The matrix is in the very inner part of the mitochondrion. That contains the nucleoid, prokaryotic ribosomes, and then we have the inner membrane space, which I'll show you on the next slide here. Now I want you to get that down. Now the inner membrane space is tough to see. It's the space between, let me get this here, this inner membrane, this is the inner membrane, the cristae of the folds here, and the outer membrane. This is the inner membrane space right here. Okay, these are the cristae. We have the matrix of nucleoid right here. Mitochondrial DNA is found within the matrix. Never saw the movie, don't ask. And we have here the prokaryotic ribosomes. Okay, beautiful. So once you get that drawn, we'll be set for today, and we're going to pick this up in the next screencast. I know you're excited.